Well, thank you so much for joining this hybrid seminar setup. I know we're waiting for a couple more people to trickle in, but we have Dr. Rabadan with us. I wanted to introduce us since we have quite a crowd in the room here. So I just wanted to introduce Dr. Rabadan, hand it over to him to talk, and then if it's all right with you, you can take questions throughout or at the end. So interactive is the name of the game. I'll be monitoring the chat for folks on Zoom and be your representative. So if you have questions, put them in there and I will for them. All right, let me just start with a little bit of an introduction. Raul Rabadan is the Gerald and Janet Carrus Professor in the Department of Systems Biology, Biomedical Informatics and Surgery at Columbia University. He is the Director for the Program for Mathematical Genomics, PMG, and the Center for Topology of Cancer Evolution and Heterogeneity. He established PMG in the fall of 2017 with the goal of bringing together scientists, mathematicians, researchers from multiple disciplines to work towards a quantitative understanding of complex biological systems. From 2001 to 2003, Dr. Rabadan was a fellow at the Theoretical Physics Division at CERN, the European Organization for Nuclear Research in Geneva, Switzerland. And then in 2003, he joined the physics group at the School of Natural Sciences in the Institute for Advanced Study. Previously, Dr. Rabadan was the Martin A. and Helen Chooljian Member at the Simmons Center for Systems Biology at the Institute for Advanced Study at Princeton, New Jersey. He has been named one of Popular Science's Brilliant 10, a Stewart Trust Fellow in 2013, and he received the Gerald and Golden Lamport Award at Columbia University in 2014. Dr. Rabadan's current interest focuses on uncovering patterns of evolution in biological systems, in particular RNA viruses and cancer. We are delighted to have you here with us today.
Thank you. I'm going to be talking about two of my favorite topics. I mean, here are microbes, viruses, and cancer. Okay? And then I'm going to tell you something about the story of how we understand how cancer behaves, what cancer is, from some of the analysis that I don't know how to, let's see here. They're moving here. Here. Okay, this is a paper. Statistician. He was working. Okay. So there is a way of I can point, or there's no pointer. So and then what he observed, there are two things that they are kind of interesting. First of all, I mean, if using the log-log scale, he was just feeding the numbers. I mean, he was just drawing by hand at the time the curves. And there were straight lines in a log-log scale. So this is he observed something that we know. I mean, older people, they get more cancers and die more from cancer than younger people. But it was a power like x to the 6, 7, something in the middle. So there was a number here. It was kind of the same rule was happening. So older people, they were getting cancers and dying. Exactly the same rule. And then the other thing is that it was the same happening in different countries. So if you just take data from the United States, you get exactly the same line in the log-log scale. So whatever was causing cancer, at the time there were several theories. Theory of cancer was the same, was universal, was happening. Older people, they get more, but they get more in a particular rate. So it's a particular mathematical formula. How many old people is going to get it at some particular time. So at the time, there were like 4 theories of cancer. I mean, probably you know already from your things. The first people looking at in the microscope in the beginning of the 20th century, they realized that some of the cancer cells, they are weird. I mean, instead of having 23 pairs of chromosomes, sometimes they have only, they have more. And all the cells in cancer, most of the cells, they have the same abnormalities. So it was the theory that was all coming from a single cell, one of our cells. One of our cells was having something weird in the chromosomes, and these chromosomes, just in the microscope, you see that this kind of proliferation. So it was a clonal theory. So it was a single clone, a single cell was proliferating more than expected, and it was one of our cells. Very little afterwards, there was these experiments here in New York. There was this scientist. He was a Rockefeller here, and then he observed that he was going to a farm, talking to a farmer, and there was this chicken that has this kind of sarcoma. Almost there. He took, and then the farmer told him, look, I mean, there are other chickens. So there is something weird here, because that means that there's something that is. So he did the following. He took from the sarcoma, he took an extract, he put it through a filter that not even bacteria can pass through. injected in a different chicken, and they was developing a different sarcoma. And then he discovered the first virus that was associated with cancer, the Raw sarcoma virus. So it was creating cancers in chickens. In the same time, in Japan, people were thinking of the idea of the cancers being produced by, and then they did experiments with rabbits where they put kind of chemical, like kind of tars in the ears of the rabbits, and they were developing this type of tumors. So there was the theory, chemical compound, virus, there was something weird going on the cell, on the processes, but developing something weird. And then later on, it was the beginning of all the atomic experiments. If you see Oppenheimer, for example, things that were all people dealing with radioactive material. You know, Colombia was playing a very important role. The whole team was pushing these rocks of uranium. So people exposed to all this material, they were developing many cancers, leukemias and many things. So there was this theory also that radiation caused cancer. There was experiments by Muller and others that they realized that, and then there was the theory of mutations. It can be caused by different, it could be chemicals, it can be radiation. Viruses, it was unclear. It was in the 70s. Very important paper. It was kind of the origin of the cellular theory of cancer. But this was kind of the beginning, and this is kind of the theory of how we understand now cancers. I mean, this is kind of cartoon. And this is this time here is running from left. This is one of our normal cells. Here is a dot here. So now we have data that our cells, they are constantly acquiring mutations. So we get mutations, kind of tens of mutations every year in all of our cells. These mutations, they do nothing. Most of the mutations, mutation there, we accumulate mutations. At some point, if the mutations happen in places in the genome, these cells start expanding. And then you have this first clonal expansion. This is a process, oncogenesis, the very beginning of the process. Then these daughter cells, they keep expanding, and then it's another mutation that happens. Yellow cross there, and then this makes even faster. And then these things, the tumor is kind of growing big enough to diagnose. Yeah? And this is the theory that, as we know, is a clonal process, so coming from a single cell. And it's mutations that could be associated with aging, it could be associated with radiation, it can be associated with chemicals, it can be associated with different things, and there's this expansion. And there's this prediction of the theory is that when you look at tumors of older patients, they have more mutations than people from younger patients. This is something that we've been sequencing like now thousands, tens of thousands of these tumors. And this is what we observe. This is from a paper. We are taking a lot of data from many different tumors, I think 10,000 tumors. And then it's kind of hard to see in here. It's very hard to see in the screen, but I mean, probably there you can see it better. Here is number of the age. There's no log state here, and then the number of mutations that you find. In particular, there are these flavors of mutations, but this is kind of the most. And then what you've realized is that in many tumors, each of these squares is a different tumor. And then in all these tumors, they have this,
that's why you need the new and improved
there is a slope here of different tumors, and then what you observe is that tumors in older people, they have many more mutations than people in younger people. This is basically the theory here. If you time passes, they have more mutations. And this is kind of the book explanation of how time happens. I mean, you have normal cells. We have 10 to the 14 cells. With some people, I mean, some people, they have mutations in DNA repair. So these mutations, they are constantly repaired. If they are not repaired, then mutations accelerate. There are some people, for example, they have mutations that they inherit from the patient, from the parents, and that they cannot repair very quickly. Mutations, they accumulate before. Okay? Exogenous carcinogens, as I was mentioning, things that are around us, things that we smoke, that we drink, that we eat. I mean, all these things, they contribute to increase the number of mutations. And this always increasing with time. As you are getting older, you have breathed more, you have done many more in your life, and all these things lead to accumulations of mutations. And that's something that we know, and this is what we observe in most of the cases. But there are some paradoxes. The first paradox that I'm going to mention is Hodgkin lymphoma. Hodgkin lymphoma is the most common type of cancer in people probably in your age. I mean, from the late teens to the early 30s. And then it's very, very common in the States. common, and then start increasing again with people that are old. Some data. Here on the left is some study that we did some years ago. This is with people here, Colombia, people in Italy. It's not very easy to sequence, some technical aspect. But here we were doing here the age of the patients. Each of the dots is the different patient. And on the y-axis is the number of mutations that we found that they are changing proteins. They are in the regions where they're kind of coding for proteins. One of the things that we observed is that the younger people, here people in the 20s and end of the 20s, they have many more mutations than people who were older. People who were, they have the Hodgkin. There was a paper from a couple of months ago from colleagues in Cornell, Weill Cornell here, and Memorial, where they did whole genome of tumors, and they observed a very similar thing. If you just take the pediatric cases and you count the number of mutations, you get around 1,000 mutations in the whole genome cases. When you're looking at older adults, the number is much reduced. It's like 25. So that's very counterintuitive, because I mean, you have older patients, they have less mutations, and what is interesting also that I'm going to show, there's some funny characteristics. So it's kind of what we see here. So what was going on here? The same thing that we can, and we did is just to color here by the patient. And red were male here. I cannot see, I don't have my glasses here. So red is male, I think, and black is female, okay? And then if you look there, what you realize is many of these patients that they are in the 20s, 30s, they are males and females. The ratio is basically in this age. But the patients are older, they're mostly male. So they are usually older guys who are getting these tumors. And these tumors, they have very few mutations. It's like having two different tumors. One is happening like in population. The other one is only happening in older people. In the older men. So this kind of bimodal distribution of tumors was already observed. This is kind of epidemiology, where here is some data from census, where you have here the incidence of cancers. In males and females, here, the left panel, say B here, that you have here is just the distribution. And there is a peak here that is not represented on the figure on the left, it's more in the figure on the right. This is very young, basically babies, but they get these tumors. And they are mostly boys. Then when you get to the 20s, it's male and female, the ratio is 1 to 1. And if you're getting people are getting older, then you have another kind of wave that is coming up, older age here. You don't see the there is no way to have a pointer here. It's just probably easier if I point to where I'm. Yeah, it's you cannot see from there. Okay, just leave it because it's going to be complicated. It's okay. So basically, they are kind of these three waves. For babies, they are people in the 20s and older people. The babies and the people, they're very old people, older people, they are usually higher in males than females. And in the middle age is basically this. And then what is interesting is that depends on the country in the world that you are. If you are going, you look here, South America, adding to the mystery here. The final factor that I want to discuss here, 20% of these, 20% of cancers of Hodgkin lymphomas here in the US, and they are associated with Epstein-Barr. Epstein-Barr is the virus that is causing mononucleosis. Everyone here is positive Epstein-Barr, and most of the people, we don't get Hodgkin lymphoma. Only a fraction of the population, they get Hodgkin lymphomas. Of the Hodgkin lymphomas, 20% of those, they have the virus in the tumor. So in tumor cells, you can see the virus is there. And it has a particular program, prescriptional program. If you just put some color here and say where is the tumors. We found the virus. What you observe is that, and what you observe is that in the 20s, it's very rare to find the ABV positive, but in the older population, it's much more common. And these are the cases that they have very few mutations. So they're EBV positive, 20% in the US, more than 50% in some countries in South America, places in the world, in North Africa. In some places, they have a higher rate of that. They're usually older patients. They have less mutations. They're mostly male. There's kind of these two routes. There's the one that have, they don't have the virus, and then there are people in the 20s, and there are people who have these tumors with the virus. They're usually older male, and also babies, but I have data to show you about babies. So this is the features that is not only happening here in Hopkins, funny features and unusual age distributions that you don't get the usual pattern of older people getting more cancer. They have a fewer number of mutations than expected. They have very few another strange feature. They are all features that happen in viral-related cancers. So what are those? Using some of the common cancers that we know, breast is usually not related to any virus that we know. Prostate is not related to any. No, lung is not, but many others we are. I mean, they are related. So roughly, 14 million new cancer cases were diagnosed. This is data from 2012. Of those, more than 2 million, they're related to like 20%. But what is interesting, it depends on what part of the world this fraction of cancer. So here in the US, it's around 5% of cancers that they are related to viruses, infections in general, agents. But if you are going to some places in Africa, particularly in the southern part of Africa, it's more than half of the yeah. So, and then there's always a question that I ask the students here. Now it's my turn. There have been seven viruses that have been associated officially to cancers in humans. Okay, which I told you one, Epstein-Barr. Okay, it's associated with lymphoma, nasopharyngeal carcinoma, gastric. Other viruses. HPV is a big one. HPV is a big collection of viruses. There are several of those that they are associated with cancer. We have now. It's great. So, what else? You know? You weren't. Okay, so then let's Hepatitis B, hepatitis C, two other viruses, very different, B and C. Okay, so we have 4 now. We have 3 to go. It's very interesting. HIV is not one of those, but it's very interesting because there's a relation between and many of these cancers they are associated with immune suppression. So HIV in people who are not treated, even if they are treated with antiretrovirals, they have a reduction in T cells, and many of these cancers that they are associated with viruses, they start. One of the reasons, for example, if you see the map of the world here, why there are so many cases here of these viral cancers in Africa is because of HIV, with AIDS. Well, very good, very good. Very good. Very famous Kaposi's. This is one of the discoveries in Colombia. So there was a Colombia team that discovered this virus in the 90s, in 1994. They look at different things that were in the cancer, and they discovered that this virus is kind of a herpes viridae. It's very similar. It's in the same kind of family of Epstein-Barr. And it was associated, it was very famous. I mean, I have a few slides here. I can talk about that. But it was identified in the 19th century. There was this doctor, Kaposi, that was traveling in the Mediterranean, and he realized that there was this kind of skin marks in older men. And then it's basically not very aggressive. And then when there was the AIDS epidemics, there was one of the kind of skin cancers that appear in people with AIDS. When people were treated, then most of those disappeared. I was talking to doctors here that they still treated some people that they're HIV positive. They don't realize it. They get kind of low T cells. Yes. What? Not in humans. Not in humans. In rabbits, I mean, in animals, there's a big collection. In mice, there are viruses that have been identified. Many of those, I mean, there have been always these theories that there are many more than this 20% that we are talking. There's a big theory there that many of the cancers could be associated to viruses, but there's no evidence. So I think there are two that they are very rare. Main one, it was discovered it was a retrovirus similar to HIV, but it's not HIV. It's LV1. It causes leukemia. It was identified in Japan, and this happens also in the Caribbean. And the final one that was from the same team discovered Kaposi's virus. It was it's called Merkel cell carcinoma virus. It was a piece of the virus was identified in this kind of very aggressive tumor. Again, people who are immunosuppressed, sometimes they develop these tumors. I mean, some of this immunosuppression, it can be because there's a genetic condition or because they're sometimes people receiving transplants. I mean, they have to be immunosuppressed for the transplant, and then they start getting some of these weird tumors. So here is the official list that you can see here. Epstein-Barr associated with Burkitt lymphoma. It was first found there. There were these kids in Africa that they have this. It's a very common lymphoma in kids. It's one of the most common in Africa, not here. I mean, here you don't find it. But it's happening in the equatorial belt of Africa. They have these kids. They have this Hodgkin lymphoma, nasopharyngeal carcinoma. They happened mostly in Southeast Asia. I mean, if you are going to South China, Hong Kong, and then going south, I mean, there's a lot of nasopharyngeal carcinoma. 100% associated with Barr. Happens also in some places in the north of Africa for reasons that are completely unknown, but these tumors, they also appear there. Here, super rare. Plasmablastic lymphomas, some of those associated with HIV. NK cell lymphomas, super interesting. And then other ones, gastric cancers. I mean, 10% of the gastric cancers, they are associated with Epstein-Barr. They are very different genetically and epigenetically. Hepatitis B, associated with hepatocellular carcinomas, papillomaviruses, associated head and neck, cervical, notice. HTLV1, the one that was mentioning in Japan, and then Merkel cell polyomavirus and Kaposi's. So this is the official list. Perhaps there was a paper last year that they identified in condition that is kind of a precancerous one. They identified another virus there, papilloma 42, that was not in the list of the previously known ones. It's a variation, but it happens in some particular lesions. So they happen in some populations. They happen for some reasons that we don't understand, and there are many, many, many questions here. I mean, characteristics associated with virus-associated cancers. What somatic genetic aberrations are more common at these things? Now we sequence. We've been collecting data from thousands of these tumors now, and then from different places in the world, and then we're the structure of these tumors, the genetic structure, the mutations that happen there. The thing that is very interesting, why they happened. I mean, nasopharyngeal, why they happened in the Asian population, and they are not happening here. Or why Hodgkin lymphoma is more common in South America, or NK cell lymphomas in native tribes here in the Americas, not in our population. So there are clearly some component that predispose to risk in different populations. It can be that it's genetic, and it could be also environmental. We don't understand all the time. Some of these tumors, they are very rare. We are collecting a lot of samples from different places in the world. And then the final thing, how this viral etiology, so the presence of the virus, responds to treatment. I'm going to mention something about immunotherapies here. Questions, I mean, before we go. This is kind of a very generic, very wide overview. Okay, so let me tell you what is the work that we've been doing. We've been collecting information from different sources. First, we have epidemiological information. So data here, global cancer statistics, where we look at places in the world where these tumors are. There are also data we are collecting on somatic mutations. Probably you know the mutations that we usually categorize in cancers. They are of two kinds. They are germline, and they are somatic. Germline, they are mutations that they are usually inherited from the body. So they are mutations that they are in all the cells in our body. So, for example, some of the mutations that if you see BRCA1, BRCA mutations in cancer, that's a predisposition risk for breast and other cancers, this is usually inherited. But most of the mutations that I'm going to describe here, and most of the mutations that you read in the cancer paper, they're somatic. Somatic means that they are acquired in the lifetime of a person. Okay? And there are different factors, as I was saying, smoking, drinking, many things that happen around, chemicals that are around. All these things contribute to create all this profile of somatic. Here we've been collecting data. I don't know, it's close to 2,000 tumors, 13 cancers of the 7 known. We look at mutations that are germline, that they're inherited. And in particular, I'm going to mention that the particular region in the genome is called HLA class 1. Probably you know. This is very important for antigenic presentation. When you get an infection, pieces of the infectious agent, they're shown through the HLA complex to these cells, and they attack. So HLA is very interesting because it's the most diverse part of the genome. So if we are taking 2 people here, there's nobody here that has the same alleles. There are 2 alleles. I mean, the classical HLA class 1 that I'm going to mention, there are 2 alleles A, 2 Bs, and 2 Cs. And if you look at 2 different A's, 2 different B's, and 2 different C's. And if you just take people around, we are different. So this has been associated to multiple. For example, it's autoimmune conditions. I mean, many people who have a very active immune system and they are getting multiple sclerosis, lupus, and all these things. There are a lot of information about HLA. It's a risk factor. Same thing for. We know there is an infection here. With COVID is arriving, and then some people are responding very well, some people they don't. So HLA is usually one of the factors that contribute to adaptive immune responses. Okay. So, and then the treatment, we are getting data from clinical trials and trying to see how immunotherapy is affecting or the presence of the virus is affecting response to immunotherapy. One of the things is fascinating. We published many papers, usually kind of pieces of papers. I mean, some of them. We work with doctors that they were in Uganda. And then we work with, this work with Kaposi sarcomas in Sardinia. Sardinia in Italy, there's an island. And then there in a particular region in the north, close to Tuscany, very high prevalence of a particular Kaposi that is usually associated with AIDS. They are not having something in the population that predispose these people there to have these tumors. So we've been collecting data from different places. We are now having networks of collaborators. They are collecting samples in Central and South America in native populations of NK lymphomas. Collaborations also with partners in Asia, collecting some of these tumors. Also in South Africa, the associated plasmablastic lymphomas. So we've been collecting samples from different places in the world. I mean, looking at different patterns of these tumors, and if there is some common structure, how these things happen. Again, it's very interesting because the epidemiology is very, very different. So here is just this is kind of more recent data compared to the 1953 paper of Norling. This is the incidence of different cancers. If you just take non-viral associated cancers, and then you just plot it here, on the y-axis is log scale, in the x-axis is not. It's just log scale. So we use the polynomial is kind of a log curve, and this is basically what you observe. Here's the first peak. They are pediatric cancers, so they are few cancers that happens before they are 8 or so, and then it starts increasing with a particular law that all the cancers, as you are getting older, you get more cancers. If you pass 80, and then the turn start curve down. So there's a point that you are old enough that you pass the risk, but you have to go beyond 80. And you reduce a bit, not much. Okay. But this is what you observe in most of the cancers. If you just take some of the cervical is papilloma associated. 100%. And what you observe is a very different curve. First of all, I mean, usually you don't get kids that have it. And then there is the big peak happens in the 30s. Usually it happens around 10 years after the fixed that is usually coming with the first. So people apply the virus. The virus takes like 10 years to start developing the tumor, and then this is what you observe with most of the cancers. Then it's going down a little bit, but just kind of very slow. Many of those, they have a very similar. This is Hodgkin. You see there's this kind of very low numbers in kids, and then going up, and then going down, and then start increasing slightly up. It depends on the country and the world. These different peaks vary. Here is 20% of those in the older population, but in other places they are much higher. So, and then that being exposed to the virus contributes to these cancers. There's this one New England paper that was in 2003 that they were taking data from, this is Sweden, Denmark, taking many people. There were people who were getting mononucleosis. So they were getting, they were getting EVV when they were in the teens usually. And then many of those, it was already observed that people getting mononucleosis, they have higher risk of getting Hodgkin lymphoma a few years later. So they did the experiment where they take many different people, and then they observed people who were taking mononucleosis and people who got the Hodgkin lymphoma. And they look at the risk, the relative risk of Hodgkin lymphoma. And then what you observe is that there was this thing. If there was the Hodgkin was EVV negative, there was no association. There was no association with mononucleosis. But the ones that were EVV positive, there was this happening usually 4 years after the mononucleosis. So they were 4 years, and then 4 years later, there's the high diagnosis of Hodgkin. What is interesting that I put here is that you noted in the paper, when we look at the data, there were 17 cases that they were kind of EVV positive of these 46. The paper, most of them, they were male. So the ones that they are kind of in this peak, they are kind of more male. They were mononucleosis and then get it. So this is kind of these funny distributions. They are usually associated because you get exposed to the virus, usually funny condition, exposed to the virus. So that happens with many of the other tumors. And then the second thing that was also interesting, you look at the male to female ratio. It's something that is very well known that in the majority of cancers, we eliminate breast, uterine, cervical cancer. In the majority of the cancers, and the rate both of cancers is higher in male than female. Higher, males will have higher rate getting cancer, and this increases with age. But this is even higher in viral-associated cancers. So here, for example, is that some of these viral-associated cancer, gastric, hepatocellular carcinoma, Hodgkin lymphoma here, head and neck, carcinomas, all of those, they are kind of associated, or a fraction of those associated with viruses. And each of those, the male to female ratio is very high, sometimes factor 6. Some of these tumors you can have, like in Hodgkin lymphoma, some of those they are viral positive, some of those they are not viral positive. And you can start comparing them. In most of these tumors, for example, in gastric here, and it's the second here on the panel on the right, the ratio of male to female in the ones that they are viral positives is factor 8 in some of the studies. Each of the dots is a different kind of study. And then while in the viral negatives, there is no difference. Answer, if you are viral, if it's negative, it's the same thing, male to female, but if it's viral positive, huge fraction. This is kind of an interesting observation. There are several hypotheses about that. The other thing is kind of this funny geographical distribution. If you take the risk of prostate cancer, of brain tumors, so you get, there are differences. So they are different. So in brain tumors, for example, the range is lower than here, for example. But then, usually quite homogeneous. Breast cancer is also quite homogeneous. There are different populations, but they are not significant at what you observe here, where you can see a population that is very tumor in South Asia, and it's basically non-existent here. The same thing here with the Kaposi sarcoma. TB8 is the virus associated with Kaposi sarcoma. And it happens specifically here in the southern tip of Africa. This is very much associated with it. This is the very high incidence of HIV. So there is this kind of funny distribution that these tumors, they happen in particular populations. Some of the tumors, I don't have the data shown here, the data. But for example, NK T-cell lymphomas, they are T cells that they are infected with Epstein-Barr. These ones, they happen in very much like nasopharyngeal carcinoma populations here in South Asia, and also in the native populations here in the Americas. So there are several populations. We are now talking in different places where they are collecting data, where very high rates of these tumors and basically absent in the rest of the population. So this is clearly a genetic component that is associated with that. And this is one of the questions that we have to understand. I mean, how we can distinguish what are the risk factors associated with these cancers. The funny thing, for example, that I found fascinating is the same agent, Epstein-Barr, causes different cancers in different people in the world. So Epstein-Barr is associated with nasopharyngeal carcinoma in Southeast Asia, where you can see here. But it's also associated with Hodgkin lymphoma in many places in the world. But not, I mean, the Hodgkin lymphoma in Southeast Asia is very low. So the same agent in different populations can cause in one cell lymphoma and in other. So very different tumors associated with the same agent. So we took up data from many different, say, like 2,000 cancers, and we've been collecting some of those. I mean, we are kind of in collaborations in different places, some of those data sets, and then we start looking at mutations. So I'm going to tell you about one particular analysis. Here, I mean, questions here before.
I had a quick.
Yes.
I had a quick question.
There's a question here. Sorry. One moment, because I don't hear you. Let me see here.
Oh.
Now?
Yes. Sorry about that. And sorry if I missed it, but out of curiosity, for the non-viral associated versions of those cancers, are they different for those regions, like overall, or is it just for the viral associated ones?
That's right. Yeah, the difference is mostly in the viral associated ones. I mean, some of those NK T-cell lymphomas, they are always associated with the virus. Nasopharyngeal carcinoma is always a virus. Some of the other ones, they are not associated with the virus. For example, Burkitt lymphoma has two different flavors. The one that is associated with the virus, and it happens only in kids in the equatorial belt of Africa. It's very clear. And then if you just take the ones that they are not ABV related, they are called sporadic. They are very low rates, and it happens everywhere. So it's usually the viral associated cancers, the ones that they are more strongly associated with the geographic distribution.
Got it. Thanks.
Question?
Yeah. So the HNO of the virus and also the
Nasopharyngeal.
yeah. So the distribution is different because one is slightly lighter in North Africa, but it's more prevalent for the HL. That's the opposite case of China, East Asia, right?
Yes.
So it's flipped.
It's flipped. I don't know. I mean, that's a very good question. The title of my virus, okay? That's right. We don't know. I mean, I don't know. I mean, it's very interesting. You know, why? I mean, the same agent is associated. As you can see here, it's just kind of. Yeah, so that's a very, it's not integrated. So in most of the cases, the genome. Papilloma, for example, it is. I mean, but Epstein-Barr is not. It's a episomal. So the virus is staying there. It's not integrated. But there is something interesting about multiple sclerosis that I'm going to mention. By the way, another thing, multiple sclerosis. Last year, this paper where they saw very similar to what you see here in the paper here in Hodgkin lymphoma, but in basically with MS, multiple sclerosis. It was the paper, it was a science paper. Yeah, the paper. So, and then data, I mean, they were looking at the risk of getting multiple sclerosis, and there was so very similar to Hodgkin. very, very messy. What is interesting that is kind of the opposite in some way. So, because multiple sclerosis happen much more in females than males. That's a high rate. These ones, they happen mostly more in males than females. So there's this idea, I have a few slides at the end that we can say, that there is this kind of sexual dimorphism in the immune system that we know. I mean, this is something that is very well known, not only in humans, but in most of the animals. So usually the immune system is stronger, and part of the immune system is females than males. So for example, in case of COVID, if you remember, in the very beginning, when people were dying and many of the very severe cases of COVID, they were mostly males than females. Autoimmune disorders are much more common in females than males. And there is this hypothesis, I mean, this is already well known, that there is kind of this sexual dimorphism in the immune responses. How this is associated to the risk of certain cancers like these ones and all these things, we don't understand the mechanism. There are several hypotheses. There are reviews talking about the hypothesis. There are important genes that, if I have time, I can discuss here in the X chromosome that it's they're associated with particular Epstein-Barr. Yes.
I was doing studies on people from African and African immigrants in a different environment to try to attribute certain things to genetics.
Yes. This is one of the classical experiments that we have with migration. Here, where they come here and then they change food, and then there have been many papers. I mean, usually many of those, for example, in nasopharyngeal carcinoma, there was one that was quite interesting. So here, if I go back to the nasopharyngeal carcinoma, there was one that we were discussing. So there were these papers that were tracking European immigration from Europe in North Africa, and they saw that there was an increased rate of nasopharyngeal carcinoma. So there is something in the geography and the environment, in the food area that is contributing to that. So there is a genetic component, and probably there is associated. Okay, let me tell you something about the genetics. So I'm going to start with one example, going back to the Hodgkin lymphoma. So as I was mentioning, 20% of classic Hodgkin lymphomas in the United States and Europe, they're infection. This number, this 20%, changes if you are going to spread them. South America is more than 50%. Depends on where you are. Then these, what is interesting is the technical complication. If you look here in this picture, what you have is that you have these cancer cells here. Sometimes they form structures that are called rich stem cells. They're kind of polynucleated cells, big cells. Up in here. In the middle of the stomach, you don't have the tumors. But these tumors, they are very, very weird sometimes. They have these kind of tumor cells that they are very dispersed. They're kind of. So from the technical point of view, it's complicated if you want to sequence them, because usually what we do when we want to sequence a tumor, part of the tumor, the DNA from these cells, you look at the genome there, the genome in different normal cells, and then you just look at somatic mutations. Somatic that happens to be in the tumor cells, but not in the normal cells. It is very complicated because, I mean, the rich stem cells, they are usually very isolated. So we are working with, and they have been from our colleagues here in Cornell. The other one, we were working with groups in Europe that have been doing laser. The laser, you just look at the rich stem cells, cut them out, and then after a long time, you collect enough cells that you can. And we've been sequencing some of those. Okay? So I'm not going to go to the details. This is from the work of two students, Eric Ladevic, now is postdoc at Memorial, and Karen Gomez, that is an MD PhD student, who they have been sequencing all these tumors. Several papers. We took data from 57 patients. We did exome and whole genome, and we look at a lot of things. We did a lot of analysis. We look at different mutations, specific mutations, mutational load, counting the number of mutations that happen in these tumors. Mutations that they are recurrent, so they happened in different patients. So you look at the patient, another patient, and the same genes they are mutated. There's suggestion that there is this gene. Mutational signatures, cells, and then HLE typing. So one of that this is in the whole genome. If you just look at the cases that they are EVV negative, that they are positive, that they have the virus. The number of mutations and the continent of having mutation is very different in both. The ones that they don't have the virus, they have many more mutations than the ones that they have the virus. It's sometimes the virus has few mutations. Okay? Both at the level of point mutation, so changing C to a T or A to a G, okay? And at the level of chromosomal rearrangement. So when sometimes you have pieces of the chromosome that are deleted or amplified, or kind of different chromosomes that they are kind of scrambled in different ways. The tumors, they are EBV negative, they are much more complex, they have mutations, they have rearrangements, and the ones that they have the virus, they have very few. This is one of the plots that if you are going to any genomic meetings, this is kind of, yes.
For the virus, because they're probably mutation, but versus if you don't have any virus, you might run into like lots of different mutations. Then
they are not so specific. Well, the hypothesis, I am going to tell you some about the specific mutations, and I can tell you some of them around. Okay. So these are, for example, genes that are related here. I mean, let me focus first on the plot on the left. This is each of the different columns here is a different patient. Okay, and they are the EBV negative on the left and the EBV positive on the right. Sorry for the people online. I'm just going here. Rows here is a different gene or a pathway that is mutated. For example, the first one here, pathway that's called JAK-STAT, it's very known. There are two different genes there. They are main drivers that they are mutated in tumors. PI3 kinase is another one, NF-kappa beta, and they're all these. So in cases that they don't have the genes, they are mutated. So they have different pathways. The tumors to develop, they have to do certain things. So they have to avoid kind of some immune response. They have to accelerate proliferation. They have to do certain things. So all these things, they usually happen to mutate. On the other hand, I mean, on the virus-related tumors, they have very few mutations, almost none, and not on these pathways. Some of these things, they are the viruses, they are bringing their own oncogenes. So in Epstein-Barr, for example, they are bringing genes, LMP1, EBNA1, some genes that we know that they are in some way they are bringing their own mutations. The virus they are bringing, the tumor they don't acquire the capability of development. So there's these two hypotheses, these two pathways. One is the mutational pathway that happens where the virus is not there, and the viral pathway. They usually have mutations, but they are much less. And I'm going to be. This is on panel B, for example, is one of the famous drivers. For panel D, GNA13 is another pathway. And all these mutations, they're specific in cases where there's a few cases where they're viruses. So this is a very general phenomenon that I mentioned that this is not only happening here, lymphoma, but if you just go looking at all the different looking at different tumors and different viruses, this is exactly the same pattern in most of them. There's one exception here. In Hodgkin lymphoma, the first 5 ones on top, the primary central nervous lymphomas, plastic lymphomas, Burkitt and gastric, Hodgkin, they are EBV. We look at the cases, we sequence, and then we look at mutations that happen in the ones there. And in all cases, except for Burkitt, they happened, the number of mutations in the negative cases is much higher than in the positive cases. In the positive cases, very few mutations. In the ones that are associated with papillomavirus, HPV, it's exactly the same thing. So it's usually in both in head and neck and in cervical cancer, the ones that they have the virus, they have the mutations that the ones they don't. The same thing with this Merkel cell carcinoma virus. I mean, cancers that they are with this virus. The ones that have the virus, they have very few mutations. So there's this kind of structure where the viruses, they bring their own machinery, and the cancers, they have to mutate the way they have two different pathways. But you can do the opposite analysis. You can look at the specific mutations and trying to see in their specific genes in the cases that they don't have the virus. And then this is becoming a little bit more technical, but I mean, when you do the statistics, you find here, this, for example, if you go to the 5th gene on the panel A here, this is a gene that is very known, it's the guardian of the genome, as you probably know. It's mutated in most of the cancers. But many of these viruses that have been mentioned, they usually have an instance for blocking p53. P53 is a gene that when they in essence, or just the cell can commit suicide if there is something going on wrongly with the cell. So one of the things, they usually inactivate p53, so they can predict they have the break. So okay. So in these viral defenses, the virus itself, they bring mechanisms for blocking the mutations. They usually happen in cases where they don't have the virus. This is very known. What is not known is what happened in cases where there's something else, the viruses they need to be mutated in order to create a tumor when they infect the cells. And in particular, there are these 4 genes, and I'm going to mention the first 2 that they are kind of very much related. The first one is the unofficial name is DDX2A and DDX3X. Okay? There are 2 genes. They are RNA helicases. So RNA, I mean, usually they can form RNA double strand, and they have to separate through many of the processes. For example, when there is transcription, sometimes you need to separate the 2 strands. I mean, sometimes you create double things, and then you have to separate. But there is splicing. I mean, you need it for export. This protein has been involved. This protein has been involved in with so there are vital functions, for example, things that is vital replication in HIV and other viruses. Host innate immune response. Sorry, I don't have my glasses, so I'm inventing here what is written. And then cancer functions, as I was saying, translation, transcription, translation, transport, splicing, many of the different processes that have to do with RNA, these proteins, they're involved. We've been working for several years on this type of lymphomas. They are called T-cell lymphomas. They're lymphomas of T-cells. These are data from 200 samples that we've been collecting from along the years. There are these 2 classical types. The first one, they are angioimmunoblastic T-cell lymphomas. Second, none otherwise specified. NK T-cell lymphomas, anaplastic large cell lymphomas. Of those, they have 2 types of those that have been associated with the anaplastic, the angioimmunoblastic, sorry, and the NK lymphoma. Very different in the angioimmunoblastic, the first ones, the viruses, they are in B cells that are around. So what is very interesting, these mutations in DDX3X, they happen in the only peripheral lymphomas that they are associated with the presence of the virus, that they are the NKs. So what is very interesting, this DDX3X, is that they are kind of these 2 homologs. Is that this is a very interesting gene, that is some of the expression. So in males, we have only 1 copy of the gene. Females, there are 2 copies. But this is one of the few genes that escapes X inactivation. Most of the genes in the X chromosome, even if there are 2 copies, one of the copies are genetically silent. So you only see the expression of one of the genes. There are a few genes that escape X inactivation, and one the X3X, where you have double the dose. And if you just look at different sets, the first thing, this is from some cancer data. This is head and neck on the right, Burkitt Lymphomas on the left. The dose in normal cells is tumor is double, females and males. So it's kind of double activity. When there are mutations, there are 2 types of mutations. There are mutations that they are inactivating the protein, they're off codons, then through a process that is called nonsense mediated decay. This protein, these RNA decays. And then what you observe is these mutations, they only happen in males. And the other ones, they happen both in males and females. And they are usually disrupting this structure here, where you have a dimer of DDX3X that binds to the double. And they are usually in the interaction. But this is a very interesting phenomenon. So the same gene is mutated in males and females, but the mutations in males, and they're associated with these things. That males, we have only one copy, and females have two, and so they cannot even if they have one. Sorry. And then, so I'm just going to go a little bit faster here. I'm going to tell you something about the different mutational signatures here. I'm just going to give you an example. Pedagogical. So this is from Merkel cell carcinoma. The Merkel cell carcinoma is the skin cancer. I mean, 50% of these cancers, they are associated with the virus. This Merkel cell carcinoma virus, the last one of the 7 that was covered, 7, I think, so it's a paper in Science. And then, and the ones that they don't have. And this, we are counting the number of mutations. And also in the mutations, you can try to see what type of mutations. Sometimes we can say, I mean, this mutation looks like it's caused by UV radiation, or it's caused by aging. So here we color the mutation of a potential origin. And one of the things here on the left, and there are different cases where you can see there's no virus there, and the right there. One of the things that you observe is that the ones that they don't have the virus, most of them, they have very high number of mutations. And the ones that they have the virus, they have very low number of mutations. The ones that they have the virus, there's a lot of yellow there. And we just look at what type of mutations they're usually with UV radiation. So I was saying this is a skin cancer. So, and then there are kind of these two different pathways of getting this skin cancer. You can get the virus, and then you don't need many more mutations because the virus themselves, they're doing many things. They are carrying their own machinery. Or you can have UV radiation. So you can be exposed for a long time to the sun and then getting a lot of UV radiation. Then you get more mutations, and then you don't need the virus to do the cancer. So there is a different problem. So looking at the specific flavor of mutations can tell you what is different. There is even speculation there could be two different cells of origin. So one is associated with the viral related one. No. Yeah. So this is related to HLA, but I don't have much time here. It's a little bit more technical. If someone is interested, we can talk later. Okay. Finally, I want to tell you something about response to immunotherapy. You know, in the last 10 years, there have been kind of a revolution of how we treat many cancers, realizing that we can awake the immune system to recognize cancer cells. They have mechanisms to hide from the immune system. Cells, they have mutations. These mutations, they can be presented with this HLA system to the external T cells. The T cells can recognize foreign, and they can attack the cancer cells. The cancer cells, they have mechanisms to block this attack by basically camouflaging using immunotherapies. Most of them, they are working by blockade. Okay? So just basically precluding that the cancer cells can hide from T cells. So they have been several in many cancers. And then what we look is that this idea that if the virus is infecting the cell, many of these be expressed in the surface of the cell. So these cancer cells that they have the virus, they're so they can be recognized by T cells in the same way that these cells. If they are not, it's because they are using the cancer cells, they are hiding using some of these mechanisms, and we know those, and this is what they are doing. So when we look at different cancer cells, here is non-Hodgkin lymphomas, Merkel cell carcinoma, head and neck, papillomas related, liver, and gastric related, and this is what is the advantage of the immunotherapy in these patients that receive it. And we see that the presence of the virus, except in the cases of Merkel, is a very good marker, especially in gastric. So in such a way that this is kind of an alternative for. This is compared to this is in gastric, for example, compared to other markers. The tumor mutational burden, the number of mutations that tumors have. The more mutations, the possibility of presenting some of those. The T cells, they can be attacked. So this is one of the markers. PD-L1 is one of these, the mechanism that they have to hide. And the presence of EVV is strongly associated, as I was mentioned, to a response to immunotherapy. So just summarizing here, I mean, cancer that develop in the context, they are very different from the other ones, including geography, in the frequency of males, age distributions. They are kind of strange. I mean, they don't follow the same rule. Virus-associated tumors, there are fewer mutations than the non-viral associated tumors. They have distinct mutational mechanisms, signatures. I mentioned only one, that is UV radiated in Merkel cell carcinoma, but in all the other ones, you can see that they are it's like the mutational has different methods of acquiring some mutation. There are some mutations that are specific to these cancers. For example, these DDXDX, the RNA helicases that they are associated with tumors. We don't understand many of these, the effect of that. There, the germline predisposition, I didn't have time to talk, but there is something like that exposes people to kind of these cancers. And virus-positive tumors may be a positive prognostic indicator of people immune checkpoint inhibitors. So it's a cartoon summary, okay? So the textbook explanation is the one on top. 80% of the tumors. You get older, more mutations. You get mutations in DNA repair, more mutations. Smoke, drink, exposed to the UV radiation, more mutations. Microbiome, we know now the microbiome can induce mutations. More mutations. So whatever you're doing, you're getting older, more mutations. Then the risk of cancers start increasing. Quite interesting. Why is X to the 6 or not to the 7 or to the 25? It's an interesting. So, however, there's a different route that follows 20% of the world. There's different infection that happens in normal cells. Usually, when there is an infection, we clear the infection. Our T cells, they are usually working on clearing the infection. But for some reason, it can be there's some genetic predisposition. It can be there's an immune condition. People they're in hospital receiving transplant, or there are people with an immune condition. All these things kind of make these T cells that they're usually cleaning very effectively. They don't do so much. And then these cells start acquiring different things, start transforming. And at some point, there are other things that happen, additional mutations and other things that make this to develop. Yes, I'm just done. Okay. So I'm here. This is the final slide. Let me just thank everyone here. This is the work of many people. I have collaborations all around the world here. I mean, some of this is the work of 3 students and a postdoc. Karen JB, that is also Mick, Theorem Physics, and Uni, that is working now. Follow up. Thank you.
Thank you so much. There is some virtual applause for you as well. I don't know if you have a few minutes to stick around. I know people have places they need to go. We are just past 2. But if folks have any last questions for you, if that's all right. Not seeing anything online. Folks online, feel free to put your questions in the chat or raise your hands. Anyone in the room?
Do you want to suppress the people? How much of your acquiring like social dependence of data from those different regions to kind of assess the effect?
That's a very good question. It's very complicated. This question. We have, for example, we have a very interesting project with groups in South Africa about some type of melanoma, acral melanoma. Very little is known about the genetics of those, and we found some interesting mutations. We look at the mutations, and some of those mutations they usually happen in the soul. And then, but some of the mutations they're associated with smoking, for example. It looks like, but what happened? I mean, how the cells were exposed to some smoke or something. So, I mean, there's some practices. I mean, usually we talk to the doctors there, but even the doctors, they don't know what is going on. So it's very hard. I mean, there are many things. For example, groups working with native populations in the Americas. I mean, there are many practices that, I mean, they are not reported, or they are not. Many of those, I mean, yeah, it's very hard. I mean, you get the genetic. I mean, this is, I mean, all the social component is very. We know, for example, that one of the factors that is contributing a lot to these cancers is this HLA homozygosis. I was mentioning we have 2As, 2Bs, and 2Cs, and they are usually most of us, they are going to be heterozygous, meaning that they are 2 different A's, 2 different B's, and 2 different C's. In families, in very small groups where there's a lot of familial related marriages, and then you see a big reduction in homozygosity. So these groups, they have very the capacity, because the 2 alleles A, they are the same, the capacity of presenting mutations or virus things is much reduced. So they get a lot of kind of diseases. And one of the diseases that could be associated is this one. See, homozygosis is important. For example, Hodgkin lymphoma is very important. But for that, sometimes it's not so easy, because you need to have kind of not only just what they are doing, but the family history. I mean, and using genetics, I mean, we can kind of try to see, I mean, how this happened, but sometimes it's not so easy. It's a very important information that we are missing. The same thing, I mean, by the way, so even in common cancers, I mean, perhaps we are drinking from this bottle and there's some chemical compound there that we don't know. I mean, so, I mean, or plastic or something that we're doing. We go into this the elevator and then you get exposed to whatever. I mean, we don't know because nobody's reporting these things. I mean, it's very hard to do that. Yes, sorry. Yes, yes, very much. I mean, many of those there were these are very well studied. Epstein-Barr is the first of the oncoviruses in humans. It was in 1957, and we know many of the roles of these genes. There was a paper also in Nature like 3 months ago that they found. So I mean, it can be another lecture about the thing. But this, for example, one of the things they have to do several things. The viruses. One of the proteins, for example, they have to mimic. They want to induce cells to proliferate, many of those. So they have induces of cell cycle, for example, many of the papilloma is very well known. They have to avoid the breaks that cells have, or the safe system. They have blocking. So many of the proteins they have to interact and block with the safe nodes. So many of these things, some of those. So for example, we don't know, for example, this is talking about mysteries. If we go back here, there are many things that are. I mean, each of those is kind of a question. The first question, I mean, why it happens in these populations and not in the rest? We have some idea of the components, but we don't know by far. I mean, environmental factors are going to be important. It's very hard to do that. Genetically, data, it's very hard. We don't know what is that. Even from the point of view of the viruses, we know the role of many of them, but some of those we don't. We know that viruses themselves, they mutate. So there's this idea that some of these mutations in the virus themselves can contribute to this. We don't have information on that. So this mutation of signatures, the different sources of origin, we don't know. These RNA helicases that I've been discussing, we don't know why they are mutated. We don't know. We're talking about people doing experiments. I mean, how this can contribute to that. We don't understand. The role of HLA, we know it's important, and we have mechanisms to do that, but we know that happens. So there are many. Okay, well, thank you. I need to come to yeah, thank you. I will go for a coffee later.